Margarida is assistant professor at the Animal Biology Department at the University of Lisbon, and she's going to talk about evolution and adaptation to dynamic environments.
Something's wrong here. Okay. Okay, thank you very much for inviting me to this conference. So I'm going to talk to you a little about the work I've been doing with my team for almost 20 years now, just in 20 minutes. So basically, what we do is that we study laboratory adaptation in one Drosophila species that is not Melanogaster. So yesterday you heard about Melanogaster with Susanna Varela. And what we study is the adaptation that populations present in the laboratory as soon as they are founded from the wild. So that is basically our frame of work. So doing this, we want to understand the dynamics of adaptation in several life history traits and also analyze the dynamics of molecular markers. So why do we do this? So basically, we use the laboratory as a novel environment. So we study adaptation to a novel environment. And that's the main target of our research. But they also started because we wanted to clarify controversies between laboratories that used populations that were longer in the lab and others that were shorter introduced in the lab. And these controversies were very, very hot when I started this research project. And finally, it is useful to analyze the impact of long-term captivity. So for problems, ex situ conservation problems, it may allow also to tackle how fast populations adapt to a captive environment, which is important also. So the main questions that we are addressing in terms of life history traits are how fast can a population adapt to a novel environment, and are there differences between life history traits in that pattern, and are there differences between populations, and we will see in the next few slides why this is important. And with the molecular markers, we want to estimate how variable is a population when it starts captivity, and how much does it lose in captivity, and also the relationship between the two types of traits. And our let's say our big question is, what is the role of history, founder effect, genetic drift, and selection during adaptive evolution, which is one of the big evolutionary questions. The material methods are very simple. So basically what we do is that we go to the wild, to a natural location, we collect flies, Drosophila subobscura, we found laboratory populations, and then periodically we assay several life history traits so that we get evolution trajectories. And we characterize also the molecular markers, which are microsatellites, to see how they change during maintenance in the lab. And then we do it all over again because we did it in several different foundations, and you'll understand why. So I'm going to shorten a little bit my talk by speaking about only some of life history traits that I've been analyzing with my team. And so I will focus on adult traits. Most of them are already, quite close related to fitness, like fecundity traits, age of first reproduction, early and peak fecundity, and starvation resistance is a trait that is not so straightforwardly related to fitness, but many studies have indicated that it's closely related to survival as a measure of somatic maintenance. And the microsatellites I will speak about 9 microsatellites. I have other studies with more, but this is enough to illustrate my results. So this will be like a story, okay? So it all started in '92 when I was still doing my PhD, but then I hiding from my supervisor. She wanted me to finish the thesis, but I wanted to check. I was doing quantitative genetics. I was a bit frustrated because the patterns were not very clear, and so I wanted to check in the phenotypic evolution of populations. So I collected flies in '92, and that's the green. So it's the green curves. What you have here is, does this work? No, it doesn't, no. So basically what you have here is, on the x-axis you have generations, okay? In the y-axis you have the traits, which is always as a difference to a control population, okay? So that we can extract environmental effects and in a sense it's a genetic quantification. So what you have here is that you see that the populations, I lost the pointer here. So the population converges to the control values because the zero line will be the value when they are no longer different, okay? So the green lines show that the populations are improving with time, converging to the zero value. And for starvation resistance, they even get better than the control values. And then in '98, I did another sample, another collection, and to my surprise, the patterns were still convergent, but quite different from the first study. So basically, what I saw was clear convergence of fecundity-related traits, and a suggestion that female starvation resistance had a more weird pattern, which in a sense I was expecting. And basically the main message was that though both the two foundations were converging, they were converging with different traits, okay? So for the repeatability issue, I didn't get repeatability. So the big question was, why do these differences occur? So at the time, I just was able to see one of two explanations. One was that there were genetic differences between the two collections in the wild, like founder effects. And another, it might be that there were changes in the laboratory environment between the two periods, between '92 and '98. And this might lead to genotype by environment interactions affecting the evolutionary rate. So then the next step was to analyze evolution trajectories of populations that were synchronous in foundation. And to have it, what I did was I made two collections, one in the same place where the others were done, which is the TW. It was done in Sintra. And then I collected in a nearby location in Rábida another population and introduced both in the lab. And these are the patterns for one of the traits, early fecundity. And you can see again that they converge to the zero values, so to the controls, but they have different evolutionary rates. So again, there were indications that there were some contingencies affecting the pattern. But then I still had a problem. Can I say that these differences are due to the natural population, to the source population? Or might it be that there were sampling effects? That is, if I went to the same place twice, I got also these differences. So this is the next question I wanted to address. So then I went again to Sintra in the Rábida in 2005. And what I did is that I made two collections in Sintra, which I called FWA and FWB, and two collections in Rábida, Nara and NARP, so that I might disentangle between sampling effects and the source population. So if the source population was more important than the samples, I would expect that these two were more different than these two relative to the differences between the two of them. So it was an hierarchical sampling structure. And taking advantage of the fact that at that time I already had data of the NW, TW, AR, and all of those, I make a meta-analysis with all these foundations to ask the big question whether there was repeatability of the process of laboratory adaptation and what caused the differences, if there were some differences, okay? So this is another way of representing my data. I will explain this so that you understand. So each graph now depicts the evolutionary rate is the slope of each evolutionary trajectory, okay? And so, for instance, for the NW, this point, this is the mean of the 3 replicates of the NW populations. And so this means that the slope was between 2.5 and 3.0 for early fecundity. So for early fecundity, NW was the one that converged quicker to the controls, okay? And the others, it's the same rationale. So basically, I analyze and compare the slopes of all 7 foundations. And I saw that there were differences between the evolutionary rates, but they differed between traits as the effects of time. For instance, in early fecundity and starvation resistance, the bigger differences were between collections, between years. And in case of late peak fecundity and age of first reproduction, the bigger differences were between locations. And I found some few effects of sampling effects. That is between the A and B of a single location, which was Arrábida, for starvation resistance. So in balance, I could say that my conclusions were that there was adaptation in general across foundations, so meaning that there was steady genetic variation in all foundations to adapt to the novel environment. But there were evolution contingencies in the pattern, in how quick they converged, how quick they adapted. I also saw that these contingencies were more drastic for less relevant traits. For instance, starvation resistance even changed the sign of the slope, and sampling effects were also present, clearly present in this phenotypic evolution. So there was a suggestion that variations could be caused by the source populations. But I was still not completely sure, because I only in 2005, I had 2 samples from each location. So then I looked at my microsatellites to see if they could tell me something more about the story. Okay, so with microsatellites, my main questions were how different are populations at the start of foundation and during the adaptive process, and do they differ on the degree of genetic variability at the start of foundation, and does this affect the evolutionary rate, and do they differ in the loss of genetic variability, and does this affect the evolutionary rate, which are questions that are important also in terms of conservation. So looking at genetic differentiation, so this is the FST, this is the PCOA, so principal component analysis with my genetic differentiation between populations. And just looking at the graph at your left, you see that all these points with an F mean the founder populations, and they are all lumped together. And by generation 3, the populations, and this is only for the 2005 data, they're already dispersed a lot. So meaning that only 3 generations cause a high differentiation between populations. And looking at the right graph, you can see now all foundations together until generation 14, and it's like an explosion of genetic differentiation. So this means that it's the lab that is causing this high differentiation. They are all together, so basically Arrábida and Sintra probably were just one DIM, one single population, and it's the sampling effects, the initial sampling effects in lab that caused this big differentiation between populations. And looking now at the loss of genetic variability, the same thing is observed. So at the start, all populations have high genetic variability and no differences, significant difference between founders. And then at generation 3, you can already see differences between the populations. And this difference is also observed in the decline of heterozygosity and allelic richness. So foundations lose differently this variability. And so I wanted to check if these differences could have an impact on the evolutionary dynamics, in adaptive dynamics. So I tried to relate the degree of variability in microsatellites, heterozygosity in allelic richness, and the slopes, for instance, of early fecundity, and there was no direct association. But when I analyzed the differences between the genetic variability and the slopes, I saw that there was some indication that those that were more variable had a higher slope. And even more importantly, I analyzed the difference between the loss of genetic variability and the slopes, and I found a negative association, meaning that population that lost faster genetic variability had a slower adaptive rate. Okay, and altogether, these data indicate that there were the initial sampling effects in the lab were the cause of the differences between my populations. Okay, so in balance, I saw clear adaptation to captivity in many different foundations, and joint analysis of the adaptive dynamics in molecular markers indicated that founder effects during the first generations affect a quick convergence across populations from the same location. So this means that the populations are very sensitive. When they are colonizing a novel environment, many things can occur even if they come from the same source population. So this means that evolution contingencies are important. So one big question that I wanted to address is, okay, so if they are so sensitive to such small differences because they are so similar at the start, can the same thing apply to differences between histories? I mean, if populations have contrasting histories, does this also cause a constraint in terms of their adaptation? Okay, so it's a different level of contingency. So to try to address, so basically the question is this: what if populations of contrasting history colonize a novel environment? Can we predict what will happen to the populations? So I have two basic expectations. If the differences in genetic background are main constraints in the adaptive process, we may see parallel or even divergent evolution in the novel environment. On the other hand, if selection is the main driving force, it may erase the signature of history, and convergence will occur. So, taking advantage of the decline of variation in Drosophila subobscura in Europe, which has a very clear decline in inversions and in body size, we collected flies from 3 contrasting latitudes in Groningen, Montpellier, and the Draga. We saw clear differentiation between them in the lab, in life history traits, body size, inversions, and so on. And following the adaptation during 20 generations, we saw a fast convergence of all populations to the same, like a same adaptive peak, which we saw also with a multivariate evolution trajectory analysis. So basically this indicates, let me just finish, basically this indicates that the fitness landscape is like a smooth fitness landscape, and they all are converging to the same adaptive peak. So it means that evolution contingencies in terms of sampling effects are not causing, in case of differences of history, are not causing different adaptive peaks between populations. So the general conclusions are that populations have ample adaptation, genetic variation to adapt, and there are evolution contingencies due to founder effects, but these contingencies does not prevent populations of contrasting history to converge to the same adaptive peak. And so, as a follow-up, we want to see if the population converge using the same molecular paths or different paths. So we'll make analysis with inversions, SNPs, and next-generation sequencing. And we will try to see if there is repeatability in the patterns by analyzing more population from these locations. So let me just thank all my team that has been working very hard in all these data that are very hard to obtain, and also international collaborations and several financial support from my center of research and from several FCT projects. Thank you.